The last ALS hypothesis I'll be discussing is the proteostasis hypothesis. Now, after a protein is made, it needs to undergo folding to become functional. Proteins will fold into a specific three-dimensional structure called the tertiary structure. Around 30% of newly made proteins are misfolded. In other words, they aren't folded into their proper shape, and therefore they need to be refolded. This refolding is accomplished by various molecular systems that assist newly synthesized proteins in folding, prevent misfolded or partially folded proteins from clumping together, help refold proteins back to their proper structures, and provide aid to repair damaged proteins. It is this maintenance of correctly folded proteins that is referred to as proteostasis, and it is needed for the cell to stay healthy and balanced. Now, the development of neurodegenerative diseases like ALS is closely linked to the abnormal accumulation and aggregation, or clumping, of misfolded proteins in specific areas of the nervous system. For this reason, disruptions in proteostasis are thought to have an important role in contributing to the development and progression of ALS. The endoplasmic reticulum, or ER, is an organelle which is a structure within a cell that carries out a specific job, similarly to how an organ performs specific functions for the human body. One of the major functions of the ER is to serve as a site for protein synthesis. The proteins made in the ER are required to undergo glycosylation, which is a process of adding sugar molecules onto a protein or lipid. After glycosylation, the proteins will be able to fold into their proper three-dimensional shape. This folding is facilitated by a system of molecular assistance commonly referred to as the chaperone system. The specific chaperone proteins involved in this process include calnexin, calreticulin, and ERP57. While calnexin and calreticulin work together to ensure proper protein folding, ERP57 will ensure formation of molecular bonds in the folding process, thereby ensuring a high stability of the protein. Now, the ER is said to undergo stress when there is an accumulation of unfolded or misfolded proteins in the ER. The ER contains stress sensors that will become activated in response to misfolded protein aggregations, leading to the activation of the unfolded protein response, or UPR pathway. The UPR will increase the size of the existing ER, thereby providing additional space to accommodate the cell's need to properly fold the proteins. The UPR will also decrease the amount of proteins that enter the ER, increase the transport of damaged proteins to the cytosol in order to be degraded, as well as increase the expression of genes that encode for chaperone proteins. Now, if the UPR pathway isn't able to decrease the ER stress, then the cells will undergo programmed cell death. Studies have found that neurodegenerative diseases such as ALS show a loss of function of chaperone proteins such as HSP70 and HSP40 due to the presence of misfolded protein aggregates that aren't able to be broken down. The two other pathways that the cell uses for degrading proteins is the autophagy lysosome pathway and the ubiquitin proteasome system, or UPS. Autophagy is a general term that refers to the process of breaking down cells and recycling their components, and it involves the formation of autophagosomes, which are double membrane sacs that enclose protein aggregates or damaged organelles. Now, this autophagosome will combine with an organelle called a lysosome, which will release digestive enzymes into the autophagosome to degrade the components inside it. There are three types of autophagy, one of which is the chaperone-mediated autophagy, or CMA, which refers to the process by which chaperone proteins recognize specific proteins and guide them to the lysosome. Specifically, chaperone protein HSP70 will bind to a specific sequence on the disease-causing protein. A receptor in the lysosomal membrane called LAMP2A will recognize HSP70, bind to it, and allow it to enter the lysosome to be degraded. Inactivation or disruption in the autophagy lysosome pathway has been commonly observed in studies on neurodegenerative diseases such as ALS. Now, the final pathway that the cell uses for degrading proteins is the ubiquitin proteasome system. This system involves an enzyme called ubiquitin ligase, which tags certain proteins with ubiquitin, thereby marking them for degradation. Multiple studies have linked ALS pathogenesis with dysfunctions in the UPS pathway, specifically how aggregates of proteins such as TDP43 trap critical enzymes of the UPS pathway, thereby impeding its function. Mice with a mutated SOD1 gene produce toxic aggregations of SOD1 protein. This mutated SOD1 protein is shown to avoid getting tagged with ubiquitin and therefore is able to escape degradation. aggregation. These accumulations of SOD1 protein can have toxic effects and impair both the autophagy and the UPS pathway, as well as destroying other cellular machinery that is used to degrade specific proteins. Moreover, other studies show that mutated SOD1 mouse motor neurons as well as spinal cord samples taken from ALS patients display a dysfunctional UPS and impaired autophagy as a result of associated protein aggregations. Now, these protein aggregations will actively disrupt the cell's quality control system by trapping and blocking the protein chaperones HSP70 and HSP40, proteasomal subunits, and ubiquitin ligases like dorphin and dynein. While both dorphin and dynein are specialized proteins in the UPS pathway, dorphin will tag misfolded proteins with ubiquitin, marking it for degradation, while dynein will transport the tagged misfolded proteins to the disposal structure. In response to this accumulating evidence of impaired UPS pathway having a significant role in the progression of ALS, researchers conducted several studies using autophagy activators to reverse the degradation of the UPS pathway. Autophagy activators such as resveratrol and trehalose have been shown to have a beneficial effect on mutated SOD1 mouse models of ALS.